Hi, my name is Nicole Dünker. I'm head of the Department of Neuroanatomy at the Institute for Anatomy of the University of Duisburg-Essen in Germany.
Hi, I'm Sebastian Nussmann. I'm a PhD student at Professor Dünker's lab at the Department of Neuroanatomy.
Today we will show you a handy system for culturing murine retinas close to the in vivo situation as organotypic retinal wholemounts. So let's get started.
Targeted ablation of genes and analysis of animal models is a classical strategy for enrolling specific retinal gene function. However, transgenic mouse models often display early lethality or suffer from severe malformations, preventing an analysis beyond embryonic or early postnatal stages. Primary cell culture is an alternative to investigate the effects of exogenously applied recombinant factors in a controlled environment. Dissociated cell culture has the advantage that the endogenous signals reaching the target cells are reduced, thereby facilitating the identification of exogenously triggered effects. However, important cell-cell interactions are destroyed in dissociated cultures. By contrast, organotypic retinal wholemount cultures provide a system close to the physiological in vivo situation, with neuronal interactions and connections still preserved. In our video article, we provide a step-by-step demonstration of the microdissection of murine eyes, including removal of lens and vitreous. After the 15-day-old mouse embryo has been killed by decapitation, the eyes are enucleated by the use of fine curved forceps, peeling the eyes from the eye socket. Two days after birth, young mouse pups are killed by decapitation. Up to postnatal day 15, the time point when mice open their eyes, the eye slits have to be mechanically opened by the use of forceps. The tip of fine curved forceps are inserted into the orbit and the connective tissue surrounding the eye is removed. As at postnatal day 2, the orbital bones are still cartilaginous and soft, it is important not to apply too much pressure with the forceps while trying to remove the eyes. 15-day-old mouse pups are killed by cervical dislocation. Afterwards, the eye slits are enlarged by two transverse cuts with spring scissors. Then the eyes are enucleated by the help of curved forceps, applying pressure to the orbit and transferred to a petri dish with PBS. Adult mice are killed by cervical dislocation and the eyes can be directly enucleated by the help of curved forceps, applying pressure to the orbit. We are going to demonstrate the peculiarities in dissection of murine eyes of different developmental stages. From left to right, murine eye at embryonic day 15, postnatal day 2, postnatal day 15 and an adult eye. In a small petri dish with sterile PBS, the surrounding eye layers have to be removed. During dissection, both forearms should rest on the bench top to stabilize the hands. The eye is turned with the lens side facing the bottom of the petri dish. When dissecting embryonic eyes, the site where the developing optic nerve will leave the eye is often covered by connective tissue, which needs to be removed to create space for entering the subretinal space between retina and pigment epithelium by the tips of very fine forceps. The sclera and the pigment epithelium are still very soft and easy to remove. Make, however, sure to completely remove the triangular, tent-like capillary plexus underneath the vitreous body together with the vitreous. This sticky plexus is difficult to remove separately from the remaining retinal cup without damaging the retina, but it will hinder the access of exogenously applied substances to the retinal layers during pharmacological manipulations. At postnatal day 2, the optic nerve at the back of the eye needs to be pinched off by forceps. The surrounding eye layers, including the sclera and the pigment epithelium, are still soft and easy to remove. Take, however, care not to damage the retina when entering the subretinal space via the optic nerve hole. The pigment epithelium is removed together with the choroid membrane and the sclera by carefully tearing to either side with both forceps. The eye cup is then turned to the lens side. The lens and vitreous grasped with forceps. And while tearing, the eye cup is held in place with the other forceps. At postnatal day 15, the optic nerve frequently keeps attached to the eye during enucleation and has to be pinched off by forceps as close to the basis as possible. The sclera is already tighter and thus entering of the subretinal space at the hole where the optic nerve has been removed, and peeling off sclera, choroid membrane and pigment epithelium becomes more difficult than at P2. Peel off the layers up to the level of the cornea, then turn the eye cup to the lens side and remove the cornea together with pigment epithelium, choroid membrane and sclera, while holding up the remaining retinal cup by the other forceps. Grasp the vitreous together with a small lens, and while tearing with forceps, keep the retinal wholemount in place with the other forceps. In the adult eye, the optic nerve, which needs to be pinched off to get access to the subretinal space, is much thicker compared to postnatal stages. The sclera is extremely firm, and penetration with forceps to enter the subretinal space will turn out to be one difficult part in dissection. Tearing off the pigment epithelium together with the choroid membrane and the sclera becomes harder and needs more controlled force. The removal of the vitreous is much easier compared to embryonic eyes, but the vitreous needs to be grasped at the sides. Do not pierce the vitreous with the tips of the forceps, as its content will stick to the forceps, hindering its removal. Before starting the culture, the hyaluronidase containing inner and outer limiting membrane of Müller glial cells need to be partially digested to facilitate the penetration of exogenously applied substances. For this purpose, pipette 200 microliter pH-balanced Dulbeckus modified Eagles medium containing 0.5 mg per milliliter hyaluronidase in each well of a 96 well plate. To transfer the retinal whole mounts without damage, the tip of a 1 ml pipette is cut a few millimeters to widen the opening, and the cutting edges are smoothened by insertion and twisting of a second pipette tip. The retinal whole mounts are transferred from the 96 well collection plate to the 96 well plate containing medium with hyaluronidase and pre-incubated for 15 minutes at 37 degrees centigrade. Then the whole mounts are transferred to a 24 well plate containing 2 ml chemically defined Dulbeckus modified Eagles medium per well. The retinal whole mount cultures are returned to an incubator and maintained for 24 to 48 hours at 37 degrees centigrade in a 5% CO2 atmosphere. (850 microliters PBS and 50 microliters bovine serum albumin are pipetted to 2 ml Eppendorf tubes with round bottom. The use of Eppendorf tubes with round bottom instead of classical conic tubes improves the precipitation of cells during centrifugation, resulting in a more stable cell pellet. After the desired culture time, the retinal whole mounts are transferred from the 24 well plate to the 2 ml Eppendorf tubes containing PBS and bovine serum albumin. The Eppendorf tubes containing the retinal whole mounts are placed into a heating block heated up at 37 degrees centigrade. 25 microliter collagenase and 25 microliters hyaluronidase are added to each Eppendorf tube, and the dissociation of the retinal wholemounts into single-cell suspension is started by 3 passes through siliconized Pasteur pipettes. Ten microliter trypsin are added and the cell suspension incubated for 3 to 5 minutes. The incubation time for enzymatic dissociation varies and depends on the size of the eyes and the developmental stage respectively. Check the stage of enzymatic digestion of the tissue by gently pipetting up and down. After a couple of minutes, 10 microliters DNase are added. After another incubation period of about 3 to 5 minutes, the cell suspension is further homogenized by slowly pipetting up and down. When the cell suspension appears homogeneous, the digestion of the tissue is stopped by addition of 10 microliters EDTA and the Eppendorf tubes are removed from the heater. One milliliter fresh ice-cold 8% paraformaldehyde are added to each Eppendorf tube and the cell suspensions are fixed at room temperature on a rotation shaker. After 1 hour fixation, the cell suspensions are removed from the rotation shaker and placed into a cooling centrifuge. The cell suspensions are centrifuged for 5 minutes at 4 degrees centigrade and at 0.2 relative centrifugal force. The pelleted cell suspension is removed from the centrifuge. The supernatant is discarded and the pellet is resuspended in 1 ml PBS containing 3 mg per milliliter BSA. After repeating these washing steps twice, the pellet is finally resuspended in 500 microliters PBS containing 3 mg per milliliter BSA 5 millimolar EDTA and 0.1% sodium acid. If an immunocytochemical staining will follow, do not add sodium acid to the resuspension buffer, as this results in loss of staining quality. A frosted end microscope slide, a cytospin filter with 1 or 2 holes, and a cytospin funnel are inserted into a cytospin slide clip, and the slide clip is closed. Make sure that the holes of the funnel and the cytospin filter are aligned. All slide clips are placed into the cytospin centrifuge rotor. Homogenize the dissociated cell suspension by gently pipetting up and down. When pipetting a 100 microliter aliquot of the cell suspension to the funnel, the tip of the pipette should reach all the way down to the bottom of the funnel. It is important not to push through the second pressure point of the pipette, as this creates air bubbles in the cell spot, which will hamper cell counts. Then the lid is placed onto the rotor and the rotor lifted into the centrifuge. The single cell suspensions are centrifuged at 700 rpm for 7 minutes. The rotor is removed from the cytospin and the lid is opened. The side clip is opened and the funnel and the slide clip are removed. Then the cell spot is marked by adding on the back of the slide while the filter is still in place. The filter is removed and the slide with the cell spot is cover slipped with DAPI in mounting medium. Alternatively, the cell spot can be immunocytochemically stained with BRDU or neuron-specific antibodies.
We just demonstrated the advantages of organotypic retinal wholemounts providing a culture system close to the physiological in vivo situation with neuronal interactions and connections still preserved. This culture system significantly facilitates the accessibility for manipulations of murine eyes. So that's it. Thanks for watching and good luck with your experiments.